Hi, my name is Rakaya Martin and David Thompson.
My name is Jordan Blaylock.
And I'm Asante Y. Murphy.
Welcome to Cell Reproduction.
What is a cell cycle? The cell cycle is a series of events in which a cell duplicates in its contents, or the things that are made within the cell, and divided to produce two new identical daughter cells. The cell cycle is broken up of three stages: interphase, the mitotic stage, and cytokinesis. Interphase is the first step in the cell cycle. Interphase is the stage when the cell carries out cellular functions and duplicates its DNA. Interphase is broken up of three parts: G1, S, and G2. 1, which is growth 1, is when the cell continues to carry out its normal functions. The cell will divide with growth signals when presence of nutrients are available. The S stage, which is the synthesis stage, is basically when DNA is synthesized or replication occurs of the DNA. At the beginning of the S stage, each chromosome is composed of one DNA double helix. Following DNA replication, each chromosome is composed of two identical DNA double helix molecules. Each double helix is a chromatid. Two identical chromatids are sister chromatids. The sister chromatids remain attached until the mitotic stage separates them. G2, or growth stage 2. The main goal of G2 is to complete DNA replication and get the cell ready for mitosis. During this stage, the cell collects all proteins needed to aid in cell division, aka in G2, protein synthesis occurs. Mitotic stage. The cell division stage includes mitosis and cytokinesis. Control of the cell cycle. A signal is an agent that influences the activities of a cell. Growth factors are signaling proteins that receive signals at the plasma membrane. The purpose of signals are to ensure that the cell cycle stage are followed by normal sequences. Apoptosis is defined to be a cell's programmed death. This is because cells start the process and continue until they are destructed. Keep in mind, when cell division increases, apoptosis decreases the number of somatic cells and loses contact with other cells.
9.2. The eukaryotic chromosome. The eukaryotic chromosome contains of a single double helix DNA molecule, but is composed of more than 50% protein. Some of these proteins are concerned with DNA and RNA synthesis, but a large majority, termed histones, play primarily a structural role. The five primary types of histone molecules are H1, H2A, H2B, H3, and H4. Surprisingly, the amino acid sequences of H3 and H4 are very similar. For example, H4 of peas is only 2 amino acid difference from the H4 of cattle. This means that few mutations in the histone proteins have occurred during the course of evolution. This also means that histones have essential functions for survival. The role of a histone in a cell is to package DNA into a small space. First, the DNA double helix is wound at intervals around a core of 8 histone molecules, 2 copies of each H2A, H2B, H3, and H4. It looks like a string of beads. Each bead is called a nucleosome, and the nucleosomes are said to be joined by linker DNA. Then the string is compacted by folding into a zigzag structure, further shortening the DNA strand. The H1 histone's job is to mediate the coiling process. This loose coil is called euchromatin, which represents the active chromatin-containing genes that are being transcribed. A more highly compacted chromosome is called heterochromatin. Heterochromatin is considered inactive chromatin because the genes contained on it are infrequently transcribed, if at all.
This is 9.3, mitosis and cytokinesis. Mitosis is the reproduction of cells. In preparation for mitosis, during interphase, the chromosomes replicate and most cellular organelles duplicate. During the S stage of interphase, DNA replicates. The phases of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, nuclear division is about to occur. The nucleolus disappears, spindle fibers begin to assemble as centromeres move away from each other, in the nuclear envelope fragment. Prometaphase, also known as late prophase, is when spindle fibers attach to chromosomes. Metaphase is when centromeres of chromosomes line up in the middle of the cell. Anaphase is when two sister chromatids are being separated and pulled to opposite ends of the cell. And telophase is when the spindle fibers disappears and a new nuclear envelope forms around daughter chromosomes and the nucleolus appears. And finally, cytokinesis occurs. Cytokinesis is the division of cytoplasm. It happens differently in both animal cells and plant cells. During cytokinesis for an animal cell, the cleavage furrow forms between two nuclei, and the contractile ring continues to separate cytoplasm until two independent, identical daughter cells are formed. During cytokinesis in plant cells, new cell walls form between the daughter cells. A cell plate is formed, and it expands outward until it reaches the old plasma membrane and fuses with that membrane, and then two new daughter cells are formed. Stem cells are special cells that can divide and produce more stem cells. They can also be used for therapeutic cloning, which produces tissues from adult stem cells or embryonic stem cells. They are also used for reproductive cloning, which is the production of new individual cells from embryonic stem cells.